Hi, I'm Shulin. I'm a graduate student under the supervision of Dr. Alexander Ross at ASU. In this video, I'll share my research process and findings in macro and nanoplastic characterization and separation. Nowadays, the increasing global plastic production become major threats to environment and humans. Macro and nanoplastics are referred to as small plastic pieces with a wide range of size from zero point 0.1 micrometer to 5 millimeter, and nano-sized plastics with a size smaller than 1 micron may lead to substantial risk to human health. They are either purposely manufactured to small sizes or formed from the degradation fragmentation, and will end up in our daily drinking water, food, or spreading in air. More disturbingly, submicron plastics have been detected in human blood and tissue, as they can be transported through the circulation system and accumulate in organs. The overall goal of my research contains several key points. First, study the AC electrokinetic effects of different types of macro and nanoplastics smaller than 1 micrometer. Second, characterize and manipulate the dielectrophoretic properties of macro and nanoplastics in human fluids. Third, establish a material reference library to address the lack of knowledge of size, shape, and abundance of macro and nanoplastics. Fourth, fractionation of macro and nanoplastics by size or type from body fluids using macrofluidic sorting devices. In this project, we apply insulator-based dielectrophoretic devices to achieve the separation and characterization of macro and nanoplastics. With this setup, we were able to study the effect of size, the effect of ionic strings on the medium, which will lead to different trapping behaviors. And we also encountered a special electrokinetic effect called concentration polarization electroosmosis, which can influence the trapping performance and can be enhanced or reduced by adjusting the frequency of electric field. We also observed the change of trapping positions when beads are absorbed by protein corona, which will also be helpful with future characterization of particles in body fluids. We also established a theoretical model to predict the trapping behaviors in our experiments based on the theory of dielectrophorese and the surface conduction, and we have approved that our observations are in great agreement with this. Besides commercial beads, we also developed a rigid protocol for generating macro and nanoplastic particles in desired size ranges from daily necessity items. This process involves cryogenic grinding, sonication, centrifugation, filtration, etc. Our resulted particles have been assessed with different techniques such as CLS and SEM. And here is an example of milled particles under SEM imaging. Future work will be conducted on various types of nanoplastics in human blood, and our material reference library will further be employed to assess NFP triggers cancer studies via gut organoid models.